Today's lecture is by Dr. Sarah Robertson, who received her Bachelor of Science degree in chemistry from Marquette University, followed by a Doctorate of Pharmacy degree from the University of Wisconsin. She completed a pharmacy practice residency at Northwestern Memorial Hospital in Chicago and a fellowship in clinical pharmacokinetics at the National Institutes of Health. For 6 years, Dr. Robertson was a reviewer in the Office of Clinical Pharmacology in the Center for Drug Evaluation and Research at the FDA. She is currently a director at Vertex Pharmaceutical in the Department of Clinical Pharmacology and Biomarkers. Please enjoy today's lecture.
Hello, I'm Sarah Robertson, and today I'll be covering drug interactions. So as an overview, I'm going to be covering the epidemiology and various categories of drug interactions, talking about the mechanisms affecting drug absorption, distribution, metabolism, and modulation of transport proteins, as well as alteration and renal elimination of drugs. We'll dive a little bit into enzyme transporter interplay and cover some complex drug interaction issues. We'll discuss a clinical interpretation and look at some examples of product labeling. I have up front some abbreviations I'll refer to throughout, so this will be a good point of reference. I'm going to be referring to the area under the concentration time curve throughout, and that is a measure of drug exposure when we look at a plasma concentration versus time curve, and we'll look at some examples of those. I'll also be using the terms substrate and modulator. So substrate just refers to the drug being acted upon or the victim, whereas a modulator is a drug that inhibits or induces, for instance, drug metabolism or a particular transport protein. So the modulator generally refers to the perpetrator of a drug interaction. So the epidemiology of drug interactions. They're not easy to quantify, particularly the clinical relevance of them. I have a couple references here. The one was a review of Medicaid records between the years of 2005 to 2009, in which it was discovered that nearly 17% of patients were found to have at least one clinically significant drug interaction. The risk, of course, increases in the elderly or patients with comorbidities or patients on multiple medications. A second publication here talks about an FDA review of NDAs approved in 2013, so fairly recent. And that review found that majority of compounds, 77%, were metabolized by a cytochrome P450 enzyme. And about the same number showed possible inhibition or induction of a metabolizing enzyme. And overall, nearly 50% or 45% had a metabolism-based drug interaction that resulted in a change of clinical significance. So the various types of interactions, we generally classify them or put them in two categories. Pharmacodynamic, which relates to a drug's effect on target, be it safety or efficacy, and pharmacokinetic, which generally refers to an impact on drug absorption, distribution, metabolism, basically impacting the concentration of drugs circulating in the body or acting at the site of activity. So some examples of pharmacodynamic interactions. We have additive combinations, and some examples there when you combine sedatives, for instance, with pain medications, or anti-seizure medications with pain medications that can cause oversedation. A beneficial pharmacodynamic interaction is that of ibuprofen and acetaminophen used together in combination to treat pain. A harmful example is zidovudine, HIV medication used early in the treatment of HIV with ganciclovir, which is an antiviral drug used to treat CMV infection. It was discovered early on in the early treatment of HIV that the two drugs in combination cause severe neutropenia. A synergistic combination is when we combine two drugs and we get a beneficial effect above that of either one. And a good example of this is aminoglycosides with penicillin used to treat gram-positive infections. A harmful combination is obviously that of barbiturates and alcohol, or sedatives and barbiturates, or sedatives and anti-seizure medications. And an antagonistic combination might be where one drug counteracts or blocks the pharmacologic effect of another. A beneficial antagonistic relationship is the use of naloxone or Narcan used to treat opiate overdose. An example of a harmful one is that of zidovudine and stavudine, again, two antiviral retroviral medications used to treat HIV. They actually compete with one another for phosphorylation, and it's been shown both in vitro and clinically that using the two in combination actually results in a lessening of antiviral efficacy. Some pharmacokinetic interactions. We generally put these in one of four categories: interactions affecting drug absorption, distribution, metabolism, or elimination. So first we'll start with absorption. So altered absorption might be impacted by pH. We know that a lot of drugs are dependent on an optimal pH for their solubility and hence permeability or uptake into the absorption. Increasing the pH in the gut, for instance using an H2 antagonist like ranitidine or a proton pump inhibitor, can result in an increased or decreased solubility of a drug. Some examples include atazanavir and raltegravir, again, two antiretroviral medications. They actually have two different responses to pH. Atazanavir absorption is decreased with their use of proton pump inhibitors, whereas raltegravir exposure is increased. So the question is, how clinically relevant are these pH-altering drug interactions? Well, there's an example with erlotinib, an anti-cancer medication used to treat pancreatic and non-small cell lung cancer. And a retrospective review of patient records actually found that use of acid-suppressing drugs in combination was associated with a significant reduction in overall survival after accounting for other factors, up to nearly 4 months. And that's in light of only a 50% reduction in AUC. Another DDI affecting altered absorption is the effect on GI motility. So use of narcotics such as methadone, we know, decrease GI motility, slow transit of medications through the gut, and can affect absorption. Whereas other drugs like metoclopramide, otherwise known as Reglan, which is used to treat nausea and other GI issues, and increases motility in the gut, can also affect absorption. So the example on the left here, we are using didanosine in combination with methadone or alone, and as you can see, methadone, again decreasing GI motility, had a significant effect on didanosine absorption and overall plasma exposure. Whereas the example on the right, we've given posaconazole, a antifungal agent, in combination with Reglan or metoclopramide, and we see a reduction in posaconazole absorption as we increase motility. Another example of altered absorption is the effect of chelation, and this is basically irreversible binding of drug in the GI tract, which can happen to certain phosphate medications, certain prodrugs. Classic examples of these are the tetracycline and quinolone antibiotics are sensitive to combination with things like antacids and dairy products, calcium-containing products. And in this example, we have a fluoroquinolone, trovafloxacin, given with either Maalox, which contains magnesium and aluminum, either separated by 2 hours, or with Maalox given just 30 minutes before, and you can see a profound effect on the absorption of trovafloxacin when you give Maalox just 30 minutes before. It's not the effect on gastric secretion of acid, however, because when you give trovafloxacin with a proton pump inhibitor, there's no effect on absorption. So it's really the chelation with the magnesium and the aluminum in this case. So we're going to move on now to mechanisms of interactions affected by drug transport. So just to orientate everybody real quickly, in the gut we have passive diffusion of drug, and every drug perfuses a little bit different and with different levels of permeability. Some drugs are effluxed out more than others by efflux proteins such as P-glycoprotein. Others rely on uptake by uptake transporters. Most commonly in the gut we think of OATP. And when we block either the efflux transport, we can see less efflux of drug back into the gut and hence increased drug levels in the blood. Same too, we could block up uptake transporters such as OATP here, which would result in increase drug back into the gut and lower levels of drug in the blood. So again, uptake and efflux transporters in the gut can be inhibited or induced, that is, increased, by modulating drugs. And these in turn can increase or decrease the bioavailability of drugs that rely on those transporters. And again, it depends largely on the compound's permeability. The lower the permeability of a compound, the more its absorption is affected by membrane transporters in general. And of the many gut transporters we looked at on the previous slide, and there are many, the two most common associated with clinically relevant drug interactions include P-glycoprotein, or PGP, and BCRP, or breast cancer resistance protein. A couple of examples of this is a well-documented interaction between quinidine, which is a PGP inhibitor, and digoxin. Digoxin we describe as a sensitive substrate of PGP, that is, small perturbations in PGP, whether it be induction or inhibition, can result in big changes in digoxin exposure. So we often use digoxin as our substrate of interest when we want to evaluate the effect of another drug on P-glycoprotein. Some clinically relevant and well-characterized PGP inhibitors include cyclosporin, erythromycin, verapamil, itraconazole, and others. Now we'll move on to drug interactions affecting drug distribution. Specifically, we'll talk first about protein binding. This is largely a theoretical DDI and generally is focused on restrictively cleared drugs. So those are drugs in which only a small fraction of drug is extracted as it passes through the liver or other eliminating organ. Because only unbound drug is cleared, you would expect an increase in the fraction unbound to lead to an increase in total drug clearance and hence a decrease in plasma concentrations. At the same time, we know only unbound or free drug is available to act upon the pharmacologic target. But because in general unbound plasma concentrations return very quickly to pre-displacement levels after a transient increase, these are rarely clinically relevant. And I have a quote here from a publication that states the general clinical importance of plasma protein binding interactions has been largely overstated. And I'll show this as an example. So this is looking at warfarin. Warfarin is often cited as something potentially sensitive to protein binding interactions because it is a restrictively eliminated drug and it's got a narrow therapeutic window. So in the top plot, we have the pharmacodynamic response, which is bleeding or prothrombin time. On the bottom, we have warfarin concentrations in plasma, both free and total. And you can see when we add a displacing drug, so that is a drug that competes for binding to plasma proteins with warfarin, we displace warfarin from proteins and you get a spike in free circulating warfarin. As a result, you also see a small increase in the prothrombin or bleeding time. It's quick and quite acute. However, because we have more free drug, we also have more drug available to be eliminated by the body. So our total drug concentration decreases, and overall those free levels of warfarin also return to pre-displacement levels. And all of this is resolved in just a matter of days. Transport proteins, as we talked about in the gut, can also play a role in the distribution of guts to other organ systems. Most notably, we think of organ systems that are highly protected, like the blood-brain barrier or the placenta or kidneys, but also organs of elimination like the liver. Inhibiting or inducing these transport proteins in these organs can affect the distribution of drug. So in the first example we have here, loperamide, an antidiarrheal medication, can cause CNS side effects. It's generally protected from getting into the CNS space by transport proteins such as, again, P-glycoprotein or PGP. But when you add an inhibitor like quinidine, that increases the CNS penetration of loperamide and puts patients at risk for CNS-associated side effects. Same to our examples with digoxin, again, this time with paroxetine as the inhibitor, which can increase the CNS penetration of digoxin. Now let's move on to metabolism. You've probably learned a bit already about drug metabolizing enzymes in the system, most notably cytochrome P450 enzymes, CYP3A4 and 5 are probably by largely the most abundant and promiscuous of the enzymes, metabolizing a large proportion of our medications. Inhibition and induction of these metabolizing enzymes, most notably CYP3A, is also the primary source of most adverse drug interactions. We can talk briefly about phase 2 enzymes, but for the most part, phase 2 metabolizing enzymes that cause conjugation are rarely associated with clinically significant drug interactions. Part of it is because the prevalence of these enzymes, they're available in such large quantities that inhibition of a phase 2 enzyme rarely translates into a very clinically relevant drug interaction. So we'll focus for the most part on inhibition and induction of phase 1 enzymes. So cytochrome P450 enzymes, again, as you've probably learned already, are most prevalent in the liver, but also in the GI tract. And when we inhibit the inhibition, we can get an increase in substrate or victim concentrations, which can result in toxicity. Usually, the inhibition occurs by competition to the enzyme site, and it's generally a quick onset and offset of effect when you add an inhibitor. But the time to the maximum effect really depends on how long it takes for the substrate drug to reach steady state. So if you consider a drug at steady state, these are the plasma concentrations that peak and wane from peak to trough over the course of a day. When you add a CYP inhibitor, you slowly start to gain a gain in concentrations, and the concentrations will increase again until a new steady state is reached. It's important, of course, if you're going to decrease the dose of a drug to bring it back down to the pre-inhibitor levels, that you consider the withdrawal of the inhibitor and the appropriate dose increase back to be able to maintain original substrate concentrations. So it's not just the onset, but also the offset of inhibition. The effect of inhibition on a substrate is, of course, greatest if that substrate relies on only a single route of metabolism. So a great example is midazolam, which is a classic sensitive CYP3A substrate. It relies only on CYP3A4, and it's very extensively metabolized. So it doesn't take much inhibition of CYP3A4 to get a very profound increase in midazolam exposure. Drugs with more than one route of metabolism, however, like voriconazole, which relies on 2C9, 2C19, and 3A4, are less sensitive. So if you inhibit CYP3A4, voriconazole still has CYP2C9 and 19 as pathways for elimination. We talked about the most common method is typically via competition to enzyme binding site, but there's another mechanism, and that's called mechanism-based enzyme inactivation. And that results when reactive metabolites form and they make a inhibitory complex. We sometimes refer to this as irreversible or quasi-irreversible inactivation of CYP. It generally results in a more profound and prolonged inhibition effect. So even after you withdraw the inhibitor, the inhibition can persist for some time due to this metabolic inhibitory complex. Some examples include the macrolide antibiotics like clarithromycin and erythromycin, as well as grapefruit juice. Here's an example of strong CYP3 inhibition by Ritonavir or Noravir, again an antiretroviral drug used to treat HIV. On the left we have triazolam, again a very sensitive CYP3A substrate. It's metabolized 100% by this enzyme and it's extensively metabolized. And when you add ritonavir, you see the profound increase in the area under the concentration time curve of 20-fold. On the other hand, zolpidem or Ambien is metabolized by CYP3A and other CYPs. So when you add ritonavir, all you see is a 28% increase in exposure relative to the 20-fold we saw with triazolam. So flipping to the other side, let's talk about induction of metabolizing enzymes. These occur by increased DNA transcription. So drugs interact with nuclear receptors, most notably PXR or CAR, PXR being responsible for the synthesis of CYP3A as well as P-glycoprotein. Interaction with this DNA transcription factor results in synthesis of new enzyme. Because it takes a couple weeks for an enzyme to be fully synthesized, it's a slower onset and offset relative to inhibition. We generally say that induction takes about a week to 2 weeks for its full effect. It also depends on the half-life of the inducer and the time to make the new proteins. This results in a decrease in substrate as we've got greater metabolic turnover due to the increase in CYP enzymes and, of course, reduction in activity of the drug. We might also see the formation of toxic metabolites. When you remove the inducer without a dose adjusting the substrate, you can get toxic concentrations of the substrate, of course. So just as we talked about with the inhibitor, you have to be mindful of both the onset and offset of inhibition and how you might be dose adjusting the substrate. Same too with the inducer, though of course the time course might be slightly different. Unlike inhibition, where if a substrate had multiple enzymes to metabolize it, inhibition of one was not so impactful. Induction can be significant even when a drug relies on multiple CYP enzymes if one of the pathways is induced. So this shows a time course of induction. Again, we're at steady state with a particular substrate. When we add an inducer, it's going to take between 1 to 2 weeks to get to a full effect until a new steady state is achieved. And this might require, again, a dose increase. And so we have to be thoughtful about where you're going to introduce a dose increase to maintain concentrations during the course and onset of induction. Here's an example of a strong inducer. Like we talked about Ritonavir for inhibition, Rifampin is a very strong enzyme inducer of CYP3A, as well as some other CYP450 enzymes. In this example, it was paired with an investigational agent being developed for the treatment of tuberculosis. This agent was metabolized only partially by CYP3A, only 20%. Yet when we add Rifampin, we saw a profound decrease in exposure of 66% up for the AUC and 85% for the trough, with the half-life shortened from 19 to 8 hours. This, of course, is potentially very clinically significant since Rifampin is another TB drug and might be something that would be paired with this investigational agent. This table shows a number of classic and common inducers and inhibitors of CYP450 enzymes. Just to point out in the strong CYP3A inhibitor list, again, we have some macrolide antibiotics. The azole antifungals such as itraconazole, posaconazole, voriconazole are classic strong CYP3A inhibitors. In the inducer side, again, we talked about Rifampin as a classic strong enzyme inducer. Carbamazepine and phenobarb are also strong inducers that are often used in drug interaction studies. And the herbal, St. John's wort, is also a strong enzyme inducer. We also have moderate enzyme inducers and inhibitors, which can or cannot be clinically relevant depending on the drug in question. And we generally think of weak inhibitors and weak inducers as not being so clinically relevant in drug interactions. So now let's talk about altered hepatic or biliary elimination via transport proteins. So again, we know, of course, that the metabolizing enzymes are inside of hepatocytes. Certain drugs pass permeably, passively into the hepatocyte and don't rely on uptake transport. Yet other drugs don't pass well by themselves and rely on transport proteins, most notably OATP1B1, 1B3, and 2B1 as some common ones for uptake into hepatocytes. If these are blocked, of course, less drug is going to get into the liver to be metabolized, and we're going to get higher systemic concentrations. A classic example of this are the statins, which rely on OATP1B1 and 1B3 to be taken up into the liver. And we'll look at a couple examples of the statins. There's also efflux proteins that push drug from the hepatocyte out into bile. These include, again, P-glycoprotein and BCRP. And if you inhibit these, you're going to have decreased biliary excretion. You may get higher systemic exposure, or you might just get greater hepatic metabolism if the drug is metabolized extensively in the liver. So this is an example of gemfibrozil and rosuvastatin. Again, rosuvastatin is a substrate for OATP uptake transporters in the liver, and gemfibrozil is an inhibitor. In this example, we see an 88% increase in exposure of rosuvastatin with gemfibrozil. So as you might imagine, after looking at that diagram of the hepatocyte, we can get some complex transporter CYP enzyme interplay. In this example, we're going to talk about atorvastatin. It's a substrate for CYP3A metabolism. It's also effluxed by P-glycoprotein and taken up by OATP1B1 and 2B1 into the liver. If you look at the very bottom first, a drug like gemfibrozil, which only inhibits OATP1B1, you see about a 35% increase in atorvastatin exposure. However, going to the very top, when you consider a drug that inhibits PGP, OATP1B1, and is a weak CYP3A inhibitor, you get a profound effect on atorvastatin exposure of nearly 9-fold. And somewhere in the middle, such as clarithromycin, that's a strong CYP3A inhibitor, but it doesn't inhibit OATP1B1, and you only see about a 4.5-fold increase in exposure. So this is where it gets really complicated to predict and anticipate what a drug interaction might cause if you know that transport and metabolizing enzymes are involved. Now we'll move on to renal elimination. Much like in hepatocytes in the gut, we have active transporters taking up drug from the blood, putting it into the kidney proximal tubules, as well as excreting drug into urine. Most notably, the OAT and OCT, as well as P-glycoprotein transport proteins, are often cited in clinically relevant drug interactions affecting renal elimination. So again, if you inhibit uptake transport, you would decrease elimination. Of course, systemic exposure of the drug would increase. Some examples of this include cimetidine, over-the-counter H2 antagonist. It's an OCT inhibitor and has been shown to increase exposure of drugs such as metformin. Probenecid is another classic example. It's an OAT1 and OAT3 inhibitor and has been shown to increase exposure of a number of drugs that are renally eliminated, including cidofovir, furosemide, and acyclovir. So let's talk about some more complex drug interactions. There are certain compounds that actually inhibit and induce enzymes at the same time. And a classic example of this is ritonavir and CYP3A. It can result in really unpredictable and potentially time-dependent effect on substrates. Whereas in the short term, after 2 or 3 days of dosing, you might see a huge spike in midazolam exposure. However, a week or 2 later, you may see a more nuanced effect on midazolam exposure as induction starts to kick in. We also talked about concurrent inhibition or induction of both enzymes and transporters at the same time. This can have a potentially additive or antagonist effect on a substrate elimination, and these are very difficult to predict as we looked at. You might have a combination of 2 inhibitors acting on different enzymatic pathways used by a substrate. So we talked about the example of voriconazole relying on multiple enzymes for elimination. If you inhibit one, there's not such a big effect, but imagine adding a CYP2C9 inhibitor on top of CYP3A. You're going to have an increased effect on exposure of voriconazole or other drugs. In addition, we have to consider the genetic polymorphisms of certain enzymes. We know that certain enzymes such as CYP2C9 can be expressed differently in different patient populations. A particular patient that is a poor metabolizer, that is, they carry a genotype conferring lower enzymatic potential of, for instance, CYP2C9, is going to rely on other alternative enzymes, and effect of, for instance, a CYP3A inhibitor might be more profound on voriconazole in somebody that's a poor CYP2C9 CYP2C9 metabolizer. In addition, we have to think about patients with altered baseline renal or hepatic elimination due to kidney or liver disease. So how do we predict these when we have a new compound in development? Well, very early in drug development, we characterize the compound extensively in vitro. So first, we analyze it. We look at preclinical or animal studies and in vitro experiments to characterize how the drug's eliminated. Is it renally? Is it through biliary excretion? Is it through hepatic metabolism? If it is through metabolism, which enzyme is responsible? Is it one of the CYP450 enzymes, one of the phase 2 enzymes? If it's renally eliminated, is there an active transport protein involved in its elimination? After we've characterized clearly how the drug is eliminated, that is, characterize its potential to be a victim of drug interactions, we also evaluate whether the drug might be a perpetrator. That is, does it have the potential to inhibit or induce enzymes or transport proteins? If we do observe some inhibition or induction in our in vitro systems, we characterize a probability of the clinical significance of it. So we look at the in vitro IC50 value, for example, if we're considering inhibition or induction, and compare it to our clinical exposures. Most notably, we look at peak or Cmax concentrations. If our peak exposure, I over IC50, exceeds 0.1 as a general rule, we might consider a clinical drug interaction to characterize how extensively our drug in development might inhibit or induce a particular enzyme. Or maybe we'll consider doing some mechanistic modeling to evaluate the potential for the clinical relevant drug interaction. When we do conduct clinical drug interactions, we often conduct them with well-characterized drugs. So, for instance, if our drug is eliminated by CYP3A and we want to know how inducers and inhibitors might impact it in patients, we'll evaluate a well-characterized strong inhibitor like itraconazole or well-characterized inducers such as Rifampin, and we'll do a drug interaction to see the impact of those two sort of token inhibitors and inducers on our substrate. If we're considering our drug to be a potential perpetrator, we're going to evaluate the effect on a sensitive, well-characterized substrate, for instance, midazolam for CYP3A, or digoxin for P-glycoprotein, or maybe rosuvastatin for OATP1B1. And depending on the results of these initial studies using probe inhibitors, inducers, or probe substrates, we'll consider if we need to do any more DDI studies. Are there any common co-administered drugs in this patient population we should consider? If the effect of the strong inducer inhibitor is profound, perhaps we need to look at a more moderate inhibitor-inducer and characterize the effect there. Should we consider drugs with mixed enzyme and transporter effects? Does our drug rely on a particular pH for solubility? In that case, we may want to do a drug interaction study with a proton pump inhibitor. When we evaluate the risk in a clinical setting for drugs that hopefully have been well characterized, we consider the therapeutic index of the victim drug. So, for example, atorvastatin is a relatively wide therapeutic window. If we see a 50% increase in atorvastatin exposure, that may not be clinically significant. However, tacrolimus or Prograf, which is used following organ transplant as an immunosuppressant, has a much more narrow therapeutic window, and a 50% increase in exposure can be clinically profound. So we'd probably consider a dose reduction or therapeutic drug monitoring in that case. In looking at a clinical profile of drugs, we also have to consider if there's other potential perpetrators involved, or is it just a single drug-drug interaction? We have to think about the time course of the interaction as we discuss. What's the onset of the inhibition or induction? When should we think about altering if we need to the dose of our substrate drug? And again, as we talked about, we have to consider both addition and the withdrawal of the potential perpetrator. Also, think about, is this DDI a class effect, or are there other options? For example, rosuvastatin and simvastatin have very different susceptibility to DDIs. They're both taken up by OATP, but simvastatin is also extensively metabolized by CYP3A, whereas rosuvastatin is not extensively metabolized. It also has a wider therapeutic index relative to simvastatin. So that might be a good alternative for somebody on a CYP3A inhibitor. And then we have to consider other confounders that may magnify it, such as organ impairment or older age. So looking at the U.S. product label, I've highlighted here in the index those sections where we find drug interaction information. In section 200 dosage administration, you'll often see a dosage recommendation in the setting of drug interactions, particularly if there's a very clinically relevant or commonly used drug that's used in combination with the product. Drug interactions might be described in warning precautions. They're certainly described in the drug interactions section. And then the clinical pharmacology section, section 12, tends to have the details of any DDI studies that were conducted by a company during development. So this is an example of a product that I worked on. This is Kalydeco, or ivacaftor, used for the treatment of cystic fibrosis. So if we look in section 7, we describe here that ivacaftor is a sensitive CYP3A substrate. It's very extensively metabolized and it relies only on CYP3A for elimination. So we did a study with ketoconazole, a strong CYP3A inhibitor, and found an 8.5-fold increase in AUC, or exposure. Based on this, we recommended a dose reduction from 150 milligrams twice a day to to 150 milligrams twice a week when we combine the drug with other strong inhibitors, and we've listed some here as examples. In addition, we did a study with a moderate inhibitor, fluconazole, and also saw a profound increase of threefold. So we have a dosage recommendation in place to reduce from 150 twice a day to once daily with moderate inhibitors. And we also have language in here about trying to avoid grapefruit or Seville oranges that might be CYP3A inhibitors. And there's also warning about the effect of strong inducers on the basis of a study we conducted with Rifampin, in which we saw a 9-fold decrease in exposure. In section 12 of the label, we've characterized the result of all the drug interaction studies what we've conducted here on a forest plot, so you can actually see the magnitude of effect of drugs on Kalydeco. We also studied the effect of Kalydeco on an oral contraceptive. So in that case, the oral contraceptive is a substrate. And you can see here there was no effect on the exposure of the progestin or estrogen component of the oral contraceptive. And lastly, I have a slide which outlines some resources and tools and references that might be useful. Thank you for your time. I hope you found that interesting and instructive. And if you have any questions, please follow up with the course coordinator.